Microbes,
like bacteria
viruses,
and fungi.
They're so tiny, you can't see them. But there are more of them in and on your body than there are stars in the galaxy.
In June 1983, a medical journal ran a letter barely longer than a paragraph. A pathologist in Perth had spent 4 years looking at slices of human stomach and kept seeing something that could not be there. Curved bacteria alive on the lining of an organ textbooks called sterile. Nobody replied. His own profession ranked the work below 56 other abstracts and declined to hear it. 11 years later, it had overturned the treatment of a disease affecting millions, and it went on to take a Nobel Prize. So, what was on that slide, and why did nobody want to look? 2 letters, 1 heading. No editorial, no cover, no press release. What the Lancet printed sat in the correspondence pages, where doctors write in about small things, and it was 2 separate letters, one after the other, under one shared heading about unidentified curved bacilli on the gastric epithelium in chronic active gastritis. The first was signed by J. Robin Warren, a pathologist at Royal Perth Hospital in Western Australia. He described curved organisms sitting directly on the inflamed stomach lining of patients, and then went further, offering a way for such a thing to be possible at all. The second was signed by Barry Marshall, a young doctor from the gastroenterology service at the same hospital. He had grown the organism in culture, and it kept appearing in patients with peptic ulcers. Splitting them preserved who had seen what. Warren had the observation, Marshall had the culture and the ulcer link, and here is what should unsettle you first. The idea in those letters was not new. It had been seen, written up, and buried at least 3 separate times already, and the last burial had been thorough. 100 years of looking away. European anatomists described spiral organisms in gastric tissue in the late 19th century. The Italian pathologist Giulio Bizzozero documented them in the stomachs of dogs in the 1890s. American researchers found them in human stomachs removed during surgery in 1940. Each sighting entered the literature and was filed as a curiosity. Then, in 1954, the door was closed properly. An American researcher, Eddie Palmer, ran what looked like the definitive study, examining well over 1,000 gastric biopsy specimens and reporting no convincing evidence that bacteria colonized the human stomach. The organisms other people kept describing, he concluded, were contaminants that arrived after the tissue left the patient. That single paper buried the idea more effectively than prejudice ever could. It was large, it was methodical, and it was wrong in a way nobody could see. Stay with this one, because it is the reason your grandparents were told to live with something a pharmacist can now cure. If a big careful study can be wrong invisibly for 25 years, everything downstream inherits the error. Subscribe, because the reason Palmer's slides showed nothing is almost absurdly small. The staining approach in general use was poorly suited to showing an organism sitting under the mucus layer tucked against the tissue surface. Palmer looked hard, in volume, and did not see what was there. A negative result from a large, careful study is one of the strongest signals a field can receive, and medicine acted on it for the next quarter of a century. By 1983 then, the observation was not new at all. It had been made, published, and formally overturned by better resourced work. It had simply never been believed, which means the wall Warren and Marshall walked into was not ignorance. It was evidence. Bad evidence, held in good faith, by people who had done their reading. What medicine knew for certain. To understand why nothing happened for years, you have to take the belief being contradicted seriously. It was not stupidity. It was a well-built consensus with real reasons behind it. The first was chemistry. The human stomach holds hydrochloric acid strong enough to strip metal. Generations of physicians were taught that nothing could colonize it, that the stomach was effectively sterile, and that anything found on a gastric slide had arrived after death or during handling. Contamination was not a lazy excuse. It had often been the right answer. The second was a model that appeared to fit real patients. Ulcers were a disorder of acid and lifestyle. Too much acid, too much stress, too much smoking, too much of the wrong food. The teaching slogan was blunt: no acid, no ulcer. And it was not fantasy, because when doctors suppressed acid, patients genuinely felt better. The third was money, and this one is uncomfortable. Acid-suppressing drugs, first cimetidine and then ranitidine, were among the best-selling medicines in the world, in the late 1970s and 1980s, and they worked in a particular way. They relieved symptoms, ulcers healed, the course ended, and a great many ulcers came back. Marshall has spoken openly since about writing to pharmaceutical companies for a modest amount of research funding and being told repeatedly that money was tight, while that industry earned enormous sums from acid suppression. Nobody needs a conspiracy to explain what follows. A treatment system, a teaching curriculum, a specialty's identity, and a commercial market were all built on one causal story. A cure that took 2 weeks and cost very little was not simply unwelcome. It was structurally invisible, which stops the question being why the profession resisted, and starts it being something harder. What kind of person keeps looking anyway, with nothing to gain and a settled literature telling him he is seeing dirt? The man at the microscope. Robin Warren was not a maverick by temperament. He qualified as a pathologist in 1967, took a post at Royal Perth Hospital the following year, and stayed for most of his working life. Gastric biopsies arrived. He examined them. He reported what was there. In June 1979, he looked at a biopsy and saw bacteria on the surface of the tissue, not floating, attached in numbers on inflamed lining. What he did next is the reason this story exists. He did not decide he had found the cause of anything. He started collecting. Over 2 years, he gathered case after case, building the association slowly. Where the curved bacteria were present, the tissue tended to be inflamed, and where they were absent, it usually was not. In his Nobel autobiography, Warren described how his spare time became devoted to studying these bacteria after that first sighting. That is the whole method in one line. Not spare funding, not a spare team, spare time. He also had a control group he never had to build. In the population crossing his bench, a large share of patients carried the organism and a large share did not. So the comparison assembled itself out of ordinary workload, one slide at a time. What he did not have was a clinician. A pathologist examines tissue after it has been removed, to connect what he was seeing to living patients, to symptoms, to who had an ulcer and who did not. He needed somebody at the other end of the process. By his own account, he had been looking for a collaborator for roughly a year and had not found one. Warren could prove the bacteria were there. He could not prove they mattered. And before anyone would let him try, he had to answer the objection every colleague raised first, which was not about disease at all. It was about survival. How anything survives down there. This is the objection that killed the idea for a century, so it is worth resolving. The stomach is not uniformly hostile. The acid sits in the lumen, the open space in the middle. The wall itself is coated in mucus, a thick gel the stomach secretes to protect itself from its own contents. And underneath that gel, pressed against the epithelial cells, conditions are very different from the acid bath a few millimeters away. That is precisely what Warren proposed in his 1983 letter. Not that the organism tolerated acid, but that it was never really in it, living in close contact with the epithelium and sheltered by the mucus above. He was describing a niche that had been invisible partly because the staining methods of Palmer's era were poorly suited to revealing anything living in it. There is a second half to the mechanism, explaining how the organism crosses the acid to reach that shelter at all. It produces urease, an enzyme that breaks down urea and releases ammonia. Ammonia is alkaline. The organism carries the means to neutralize its own surroundings, generating a small buffered pocket around itself as it burrows into the mucus. Once that is understood, the impossibility dissolves. The stomach was never sterile. It was sampled the wrong way, described in the wrong terms, and defended by a slogan. But mechanism alone convinces nobody in medicine. And in 1981, Warren still had a drawer of slides and no clinical partner. Then, a registrar with no particular interest in gastroenterology or microbiology was told he needed a research project and was pointed down the corridor. The first person who was interested. Barry Marshall was 30, working through internal medicine training at Royal Perth Hospital, where junior doctors were expected to produce a small piece of research each year. His earlier attempts had gone nowhere in particular. A consultant offered a few options, one of which was to go and see the pathologist who had been hunting for a clinician. Warren later said something about that meeting that carries more weight than any technical detail in this entire story. He described Marshall as the first person who was actually interested in what he was doing. Sit with that sentence, because it holds two men and two kinds of loneliness. Warren had spent 2 years accumulating an observation nobody wanted, in a specialty where being interesting is not the job. Marshall was a junior doctor with a box to tick and no obvious future in either field, which meant he had almost nothing to protect. He was not a believer walking in. He was willing to find out, and that turned out to be rarer and more useful than conviction. They set up a proper study, examining gastric biopsies from around 100 patients undergoing gastroscopy. Marshall supplied the clinical side, the histories and endoscopy findings and specimens. Warren read the pathology. By late 1982, the numbers were in, and they were not subtle. The bacteria tracked with gastritis. More striking still, among the patients with duodenal ulcers, the organism was present in effectively all of them. Getting there had required something the pair had failed at for months. The organism refused to grow. Standard laboratory practice was to incubate culture plates for 2 days, and then discard them, and attempt after attempt produced nothing. The break came at Easter 1982, and it came by accident. Because of the long holiday weekend, the plates sat in the incubator far longer than the usual 2 days. When somebody finally read them, colonies had appeared. The organism was simply slower to grow than the routine allowed. For months, the answer had been thrown in the bin on schedule, on time, every time, by a laboratory doing exactly what it was supposed to do. That is the hinge of the whole story, and it is not a story about genius. It is a story about a protocol. Consider what rests on that accident. Without a culture, the pair had slides and an association, which is what a century of earlier observers had, and what had been dismissed every time. A culture changes the category of the claim. The organism can be grown, named, tested against drugs, and given to something else. Marshall did exactly that, working out during 1983 which agents it was vulnerable to, starting with bismuth. So by early 1983, they had an observation, a culture, a mechanism, a 100-patient dataset, and the first hint of a treatment. What happened next was the worst year of the entire project. The year the door closed. They did the ordinary thing first, submitting an abstract to their own national body, the Gastroenterological Society of Australia, hoping to present at a meeting in Perth. It was rejected. The rejection letter has since circulated widely because the arithmetic in it is almost too neat. Of 67 abstracts submitted, the committee explained, they had been able to accept 56. Marshall's was not among them. He has said since that this placed the work in the bottom fraction of everything offered that year, and he has compared the idea of a germ causing ulcers to telling gastroenterologists that the earth was flat. Terry Bolin, the gastroenterologist who chaired that selection process, spoke about it publicly in later years, and has been identified in accounts of the episode as the man who turned down what became Nobel-winning work. He did not hide from it. And in fairness, the abstract was competing against 66 others in a field where the acid model explained the data adequately, submitted by a registrar contradicting the textbook. Nobody in this story is a villain. The system did what the system was designed to do, which is filter aggressively in favor of the established explanation. It simply happened to filter out the correct answer. So they went sideways. If the gastroenterologists were not interested, the microbiologists might be, and they had no stake in the acid model. Marshall was put in touch with Martin Skiro, a British clinical microbiologist who knew the bacterial family the organism resembled. Cultures were posted to him. He grew them, and he became interested. In September 1983, Marshall presented the Royal Perth findings at the Second International Workshop on Campylobacter infections in Brussels. And the reception was the inverse of Perth. European microbiologists took the data seriously and began following it up in their own laboratories, which makes 1983 a strange, split year. The observation reached print in June, readable by everybody and acted on by nobody. The body that should have amplified it declined to hear it. A workshop on another continent took it up instead. And when the pair submitted a full clinical report to The Lancet, the editors hit a problem that says everything about where the field stood. Willingness to publish was not the obstacle. Finding reviewers who took the subject seriously enough to assess it was. The full paper appeared in 1984. It still was not enough. To understand why Marshall then did something no ethics committee would have approved, you have to understand what an ulcer meant for the person who had one. What an ulcer meant before 1990. Strip away the abstraction, because the stakes here were physical. A duodenal ulcer was not an episode. It was a condition you were assigned for life. Acid suppression brought relief, so patients took medication and improved. Then the course ended, and a substantial share relapsed, which meant going back on the drugs, often indefinitely. Around that sat a scaffolding of management, dietary restriction, bland food, instructions about stress, about smoking, about how the patient was living, which quietly located the cause inside the patient's own character. Ulcer disease was something you were told you had brought on yourself by being too anxious, too driven, too badly behaved at the dinner table. And when medication and lifestyle failed, the answer was surgery, on the same logic as the drugs. If acid is the problem, reduce the capacity to make it. That meant operations to cut the nerves driving acid secretion, and in severe cases, removal of part of the stomach. People lived afterwards with a permanently altered digestive system, sometimes with lasting consequences for how they ate, absorbed nutrients, and felt after meals. That was the standard of care. Not a failure of care, the standard, delivered competently by good doctors following the best model they had. So the argument was never academic. Every year the causal question stayed open. People were medicated indefinitely, told to manage their temperament, and occasionally cut open for an infection that could have been cleared in a fortnight. Marshall knew that. He also knew his evidence had a hole no amount of correlation could fill, and that filling it required a healthy human being. 10 days in July. The hole was sequence. The slides and the cultures showed the bacteria were present where disease was present, and every critic could still argue the reverse, that a damaged, inflamed stomach was simply an easier place for opportunistic organisms to settle. Closing that requires a healthy subject, deliberately infected, who then becomes ill. Attempts in animals had failed. There was no animal model. In late July 1984, Marshall solved the problem with the only healthy subject he had unrestricted access to. He was 32, then working at Fremantle Hospital. The details come from his own accounts and from the paper published the following year in the Medical Journal of Australia. It began with a baseline. An endoscopy confirmed that his own stomach was healthy. No inflammation, no damage, no sign of the organism. That step matters more than the drama that follows, because without it, the exercise proves nothing. On the morning itself, he took a dose of cimetidine to reduce his stomach acid, reasoning it might give the organism a better chance of establishing. A colleague prepared a culture taken from a patient with indigestion, suspended in a small volume of broth. He drank it in one go. Then, nothing. For 2 days, nothing at all. Imagine sitting with that particular silence, having just swallowed something you believe causes disease, waiting to find out whether you have proved your case or ended your career for no reason. On the 3rd day it started, nausea first. Then his breath turned foul, and here is the detail that makes the episode human. His wife noticed, his mother noticed. Both commented on it before he admitted what he had done, because he had not told them, and he had not told his superiors either. He has described the arrangement around him afterwards as a matter of not asking and not telling. Then came the vomiting in the mornings, the way his patients described it. Around day 10, he went back for the second endoscopy, and the stomach that had been cleaned 10 days earlier showed severe gastritis and damage to the lining, with the organism he had swallowed colonizing the tissue. He treated himself with antibiotics and cleared the infection. He has said, without performance, that he did not expect to become as ill as he did. Here the skeptics have a real point, and it deserves stating rather than burying. A single subject is not a trial. There was no control, and pre-medicating with an acid blocker means the experiment did not show the organism could establish itself in a completely untouched stomach. Marshall himself never claimed the drink settled the science, and the case never rested on it. What made the argument hold was that he was also curing people, treating stubborn duodenal ulcer patients with bismuth and an antibiotic, watching most of them improve and watching the ulcers stay away once the organism was gone. That was the thing the acid model could not match. Suppress acid and the ulcer heals and returns. Remove the bacteria and, in most patients, it does not come back. Local doctors noticed and started sending more patients, which left one question, and it had stopped being a scientific one. The evidence had been in print since 1983. The next decade is where this story gets difficult to defend. The name and the thing nobody wanted to say out loud. The organism was first grouped with the bacterial family it superficially resembled, then renamed once analysis showed it did not belong there. By 1989, it carried the name it has now, Helicobacter pylori, the helix in the front half, describing the spiral shape Warren had been pointing at on his slides for a decade. The clinical turn came in the 1990s. Independent groups replicated the association. Treatment trials moved from single hospitals to multi-center studies. In 1994, an American National Institutes of Health consensus panel concluded that the organism was central to peptic ulcer disease, and that ulcer patients carrying it should receive antibiotics. In the same year came the finding that raises the stakes of everything above it. The World Health Organization's Cancer Agency classified the organism as a carcinogen, on evidence linking chronic infection to stomach cancer. That is the fact that deserves the longest pause. Gastric cancer has been one of the leading causes of cancer death worldwide for decades, with poor survival when it is found late, which it frequently is. And the 1983 observation, printed in a correspondence column and ranked below 56 other abstracts, opened a route to preventing a share of it by treating a common infection. The hedging matters here, and the sources hedge it. Most people carrying the organism never develop stomach cancer, and never develop an ulcer either. Infection alone does not determine who becomes ill. The classification established a contributing cause of a major lethal cancer, not the whole explanation of it. Set against that, the treatment is almost insulting in its ordinariness. A short course of antibiotics with acid suppression, 1 to 2 weeks, high cure rates. That is what replaced lifelong medication, dietary rules, repeated relapse, and stomach surgery. Even then, the change came slowly. There was a stretch in the 1990s where the cure existed, was published, was endorsed by expert panels, and still was not reaching a large share of patients because prescribing habits formed over 20 years do not turn over in a season. Surveys in that period repeatedly found substantial numbers of physicians still managing ulcers primarily with acid suppression. In October 2005, the Nobel Assembly at the Karolinska Institute awarded the Nobel Prize in Physiology or Medicine jointly to Barry Marshall and Robin Warren for the discovery of Helicobacter pylori and its role in gastritis and peptic ulcer disease. The citation named the bacterium and the disease. It did not need to name the resistance. 22 years separated the correspondence pages from the prize. Warren was in his 60s by then, having spent most of the intervening period doing the same unglamorous work in the same city. What the 1983 letter actually proves. The evidence was never hidden. From June 1983, it sat in one of the most widely circulated journals in medicine, readable by anyone with a library subscription, describing an organism, a location, an association, and a plausible mechanism for how it survived. Nothing about it was secret or hard to obtain. What kept it dormant was not concealment. It was the sorting logic of a profession that already had an explanation it trusted, backed by a large, careful, wrong study from 1954 that everyone had read. Most of the counter-arguments were reasonable at the time. The stomach really is an extraordinarily hostile place, and doubt about colonization was defensible chemistry, not prejudice. Association really is not causation. Most carriers never develop an ulcer, which remains true and still means infection does not by itself explain who gets sick. Every one of those was a fair question, and everyone was eventually answered by work rather than argument. But some of the resistance was not about evidence. An abstract was ranked below 56 others in a year when it contained the answer to a disease affecting millions. A journal struggled to find reviewers willing to engage with it. Funding requests were declined by companies earning enormous revenue from treating the symptoms of the same condition. Robin Warren did not need to be right about everything in 1979 for that observation to matter. He did not have causation, and he did not have the cure. He had one thing, a refusal to explain away what he could plainly see through a lens because the literature said it should not be there. He collected instead, case by case, for 2 years, until somebody with clinical access walked in and asked what he was working on. So, the discovery was not a flash of insight. It was a person declining, quietly and repeatedly, to file something correctly. If that reframes how discovery works for you, subscribe, and leave a comment on this one. What is the modern equivalent, the observation being ranked in the bottom 10% right now because it contradicts the model everybody trusts? There is another episode on the ulcer story's strangest chapter on screen now, the decade when the cure existed and almost nobody prescribed it.